Okay, so the other thing that you might want to get out while we're doing this is your energy chart that we did yesterday. The respiration part, it'll help you understand where all these molecules that I'm talking about come from. All right, so I would maybe have my energy chart out to the side too. And from that energy chart, you can answer this first sentence. What is the purpose of respiration? So you're converting energy from food into usable energy for the cell. What is the usable energy? ATP. And most of the time, when we're talking about the food energy, we're talking about glucose. But there are other sources of energy that your body can use, and I'll briefly explain that in just a minute how that works. But we're taking our food energy and we're making it into ATP. There's two ways that this can happen. When it happens in the presence of oxygen, when it happens with oxygen, it's called aerobic. Because aerobic means oxygen. And when it happens without oxygen, it's called anaerobic. Do you guys remember when you put an or a in front of something, what does it mean? Like not or without. Like abiotic, not living. So both processes are going to start in the cytoplasm with glycolysis. And I'll explain what that word means in just a minute. But the reason you have a page pointing this out is this is where all organisms start. Every living thing on Earth can do glycolysis, and it's how we begin to break down glucose to harvest energy. Now remember, what cellular respiration is going to be is a series of steps that help us break down glucose and harness the energy. Do you remember why we can't just break it all down at once? Too much energy. The cell will explode. So everything we're doing is little baby steps to harvest as much energy as we can without exploding the cell. We've got to do it in steps. The next page is background information that I think is helpful to understand this. You will not learn this in detail until chemistry next year. Okay, but cellular respiration is powered by something called oxidation and reduction reactions. And it's all about transferring electrons. Remember, electrons are held in what part of the molecule? The bonds or the nucleus? The bonds, okay? And we talked about yesterday how making a bond stores energy and breaking a bond releases energy. It's all about the electrons. When you lose electrons, you're losing some energy. When you gain electrons, you're gaining some energy. Bonds breaking and making is electron transfer. Okay, and we call that oxidation and reduction. It's gaining and losing electrons. The symbol for electrons that I'll use from now on is this. Okay, E with a negative. And that's helpful because electrons are negatively charged. Oil rig is just an acronym to help you remember what each reaction is. Oil oxidation is losing electrons. So oxidation is losing electrons. Okay, so think about this. Would you lose electrons, or does a molecule lose electrons when it's formed or when it's broken down? When it's broken down, because that's when you're releasing the electrons. So when we say a molecule is being oxidized, it's being broken down. Okay, so oxidation is losing electrons. Here's where it gets a little confusing, and I'll explain why. Reduction is gaining electrons. Now, the reason that's hard is, to me, reducing is the opposite of gaining. Do you guys get what I'm thinking with that? The reason it's called reduction is because what's being reduced is the charge. Remember, electrons are negative. So if a molecule gets more electrons, it's becoming more negative. It's reducing its charge. That's where the reduction part comes from. When you gain electrons, you become more negative, the charge is reduced. If you don't want to have to think about it too hard, just remember oil rig to keep yourself straight. Okay, this is going to be all about energy carriers and electron carriers again. If I say energy carrier, let me say that again. If I say energy carrier, electron carrier, or electron acceptor, it's all the same thing. It's molecules that can store energy in the form of electrons. Which two did you learn in photosynthesis? ATP and NADPH. For NADPH, I want you to underline the P, and I'll tell you why in just a minute. Okay, and then your other one is ATP. Our energy carriers in respiration, we're still going to have ATP, but we're going to add two new ones, NADH and FADH2. We lost our P. Why did I have you underline P and the one that's for photosynthesis? To help you remember, okay? The one that's in photosynthesis has a P in it. P for plant, P for photosynthesis. Okay, the energy carrier for respiration is just NADH. And they're just different molecules that have evolved to carry energy. Don't worry yourself too much with the names. Just learn to identify the names. You don't have to memorize it. You just have to recognize it if it's in a question, okay? So these are our energy carriers. Out of those energy carriers, which one is the energy carrier that can be used for all cell processes? ATP. And remember, a recent process that we've discussed that requires energy is active transport. Okay, now we are going to work through a series of steps. Remember, photosynthesis is just one equation, right? It's a summary equation, but how many steps is it actually? 2, or I should have said stages, but light dependent and light independent, right? Respiration is the same way. That big formula is a summary of 4 steps. So we've got to learn each step. And remember, the reason we're taking steps is because glucose has too much energy, and I've got to break it down a little bit at a time. I made this diagram because most of the diagrams for glycolysis are a little advanced for what we're trying to do in intro bio. So the oval on the outside is your cell membrane. Inside is your cytoplasm. And what organelle is this down here at the bottom? That's your mitochondria. So we are not in the mitochondria yet. Okay, we are doing glycolysis in the cytoplasm. Does anybody know what the what lyse means, or lysis? Break. Like Lysol is called that because it breaks open, it breaks down bacteria. So lyse means to break down. And then glyco, what does that sound like that you've learned already? Glycogen. So glyco refers to carbs, specifically glucose. So this word literally means carbs breaking down, carbs broken. Okay, look at your energy chart, or look at your diagram on this slide. What carb is being broken down? What's going in? Glucose. Glucose is going in. Glucose is a 6-carbon sugar, and you've got a worksheet that shows you this. But glucose is a 6-carbon sugar. If I break it in 2, how many molecules do I have? 2, and they each have how many carbons? 3. Okay, so that's what pyruvate is. It's 2 3-carbon molecules. I've broken my glucose in half. When I broke my glucose in half, was that oxidation or reduction? It was oxidation. So glucose lost electrons. It lost those electrons to energy carriers. Can you tell from this part of the diagram which 2 energy carriers collected those electrons? The ATP and the NADH now have the electrons from glucose. All right, but I'm not done yet. I can still squeeze some energy out of pyruvate. There's still energy to be had, but remember, I've got to do it in small little steps. So this is the first step. The other thing I want to tell you, like when you take AP biology, I'll show you the formula for glycolysis. It's this long. It's huge. Okay, so this is glycolysis simplified for you, but really it happens as a 12-step reaction. Okay, but we've simplified it for right now. It's 12 steps, And at certain parts in the reaction, food that is not glucose can enter. Energy that is not glucose can enter. Sucrose, which is also a sugar, can enter. Proteins can enter, and fats can enter. So glycolysis doesn't only break down sugar. You can break down other energy sources as well, but in this class, you're only responsible for what? Glucose. Okay, so look at your chart. That is one of your reactants, right? We've dealt with glucose. Glucose is broken down in glycolysis. What reactant is left? Oxygen. We are not going to use oxygen until the very, very end. Okay, so this is our first step, and it's happening in the cytoplasm. Okay, it does not require oxygen yet. Now, there are two things that can happen at this point. If there's no oxygen, cells that are able can do a process called fermentation in the cytoplasm. Okay, your muscle cells can do that. Other parts of our body cannot. That is Thursday's lesson. Okay, but this is what we're going to focus on today, in the presence of oxygen. So you can outline this arrow. When we have oxygen, it's going to go to what organelle? The mitochondria. Okay, where we are going to do aerobic respiration. Right, so that's the first step, glycolysis. It does not happen in the mitochondria. It happens in the cytoplasm. How many steps do I have left? 3. Remember, I told you it's a 4-step process. So we've got 3 steps to go before we're done. Okay, the next goes in. Look at your picture. Glucose. Now, the other 2 things that go in are NAD plus and ADP. But you guys don't have to write that down. That makes it a little cumbersome. But, you know, your energy carriers have to come from somewhere. The prefix for those energy carriers are ATP and NAD+. But that's not our focus right now. And then what comes out is NADH and what else? How many ATP? 2 ATP. And 2 NADH as well. But just ATP is the one I want you to remember the number for. Just to keep things simple for right now. Simple as they can be. It can't be too simple. It's not. That's why I said simple as possible. It is not simple. It's hard. Okay, so now we are inside the mitochondria. And I have 2 steps on this slide. Originally, when I made this slide, I left the link step off. I'll explain what it is in a second, because I thought it would help you. But a lot of the diagrams that you see have acetyl-CoA on them, so you need to know where that comes from. Pyruvate enters the mitochondria, so I'm in the mitochondria now. Okay, but before it can actually enter the Krebs cycle, this is your second step. There's a step called the link cycle that's not really, or the link step. It's not really in your curriculum, but I want to tell you so you know. In link, pyruvate is converted into acetyl-CoA. And acetyl-CoA goes into the Krebs. So it's just one more conversion step. What do you really need to know about that? Acetyl-CoA is what's left over from breaking down what? Breaking down pyruvate, which is what was broken down from glucose. So acetyl-CoA is the energy that I have left. It's what's left of my glucose. I can still squeeze some energy out of it. And that's what the Krebs cycle does. Why do you think it's called Krebs? It's the guy who invented it again. Okay, so Krebs is just the guy who invented it. This cycle represents that there's a series of reactions happening. Okay, that's deep, deep chemistry. So we're just going to focus on what's made. Acetyl-CoA is going to give up its electrons. So is it being oxidized or reduced? Oxidized. We're going to break down acetyl-CoA. So this is my final step, breaking down glucose. Okay, I'm going to make 3 energy carriers. I'm going to make some more NADH that's carrying electrons. I'm going to make 2 FADH, which we haven't had yet. And then what's my third energy carrier? ATP. Okay, so look, FAD plus, NAD plus, and ADP, are they being oxidized or reduced? They're reduced because they accepted the electrons from acetyl-CoA. Okay, so they're holding the electrons now. You can underline or circle the product if that makes it easier. The little arrow represents that, FAD plus is what goes in, FADH2 is what comes out. What do I need you to know about them? They are carrying what? Energy in the form of electrons. Okay, the ATP can be used by the cell, but the FADH2 and the NADH we're going to use for something else in just a minute. So I've squeezed as much energy, as much electrons out of the glucose that I originally had. I'm done with it. I've done as much as I can with that carbon molecule. So look at your products. When I break down acetyl-CoA, remember acetyl-CoA was made from glucose. So it's a carbon-based molecule. Look at your products. What carbon-based molecule is released in this step? Carbon dioxide. You've got 3 products. You've got ATP, water, and you've got carbon dioxide. CO2 is released in this step. And the reason why, remember, acetyl-CoA is what's left of my carbon molecule. And when I break it down again, I'm releasing carbon. So that carbon's released in CO2. Okay? So your main point, this should really be main points because there's more than one. Energy carriers are made. I make NADH, FADH2, and 2 ATP. Because I broke down a carbon-based molecule, because I oxidized a carbon-based molecule, carbon dioxide is also released in this step. Okay, look at your chart, your energy chart. How many molecules of ATP are you going to make from respiration? 36. How many have I made so far? 2 from Krebs and 2 from glycolysis. So I've only made 4. So I still have 32 ATP to go. And that's what my energy carriers are going to be for. They're going to power the electron transport chain. And you also had an electron transport chain in photosynthesis, but I kind of breezed over it a little bit so that we could talk about it here. Okay, so now we've done glycolysis, step 1, link, step 2, Krebs, step 3, 4, electron transport chain. So this is my grand finale, okay? This is the purpose. This is where I'm going to make my most energy. You have, if you go all the way back, here's your mitochondria, right? We are now in this space right here. Can you guys see what I'm outlining? So we are between 2 membranes. We are between the inner and the outer membrane. So this is a closed space with membranes on both sides. All right? The problem with this picture is it's only showing you one. So I'm going to kind of draw in your other one. Okay, you also have a membrane here. Okay, in this space in between. Okay, you don't have to draw it in if you don't want to. But this is the inner membrane space because I have 2 membranes. Okay, which means I can build up a gradient because I have 2 membranes. Over here, start over here. This is my electron transport chain. The red proteins are my electron transport chain. You guys ever played hot potato in PE? Recess, is that better? Did you play it in recess? Okay, what you do is you're passing usually like a ball, and you're passing it as fast as you can, right? And that's why it's called hot potato. Well, electrons are like hot potatoes. They are full of energy, and they have to be moved quickly between each protein. So electrons are going to be dumped, and they're going to be passed through this electron transport chain. Look right here and tell me, where do your electrons come from? FADH2 and NADH. So this is where you're going to use your electron carriers. Okay, they're going to dump off their electron carriers, and they're going to travel through the electron transport chain. What is the charge of electrons? Negative. Negative attracts positive, right? So the negative attracts hydrogen, and it pulls the hydrogen into the middle of the mitochondria, into the inner membrane space. And I'm going to build up a gradient, which means I have more hydrogen where? On the inside or on the outside? Ooh, that's ugly. Okay, yeah, but on the inside. Okay, and you know molecules move from high to low, but there's a problem. Hydrogen is charged, right? So what does it need to move through? A protein. That protein is ATP synthase. The hydrogen is going to move through the ATP synthase, and it's going to move so quickly that it spins, and so like kinetic energy, and that spinning is what allows you to make ATP, a lot of ATP, 32 to 34 ATP. Okay, so that's part of it. The other thing is look at your chart. Which reactant have I not used yet? Water's made. Water's a product. I haven't used oxygen yet. This is also why I need oxygen. Y'all, electrons contain a lot of energy. They can't just bounce around free in the cell. They have to end up somewhere. Okay, so oxygen is your final electron acceptor. It's going to take that electron. Okay, but remember, electrons are negatively charged and electrons form bonds, right? That electron is going to form a bond between oxygen and hydrogen. Look at your products. What product is just oxygen and hydrogen? Water. Okay, so oxygen has to take the final electron because that electron has to go somewhere. It can't bounce around in the cell. Okay, so oxygen accepts the electron. But when it takes an electron, it also takes what? Hydrogen. Okay, and when the oxygen accepts the electron and takes the hydrogen, what product do you make? Water. Okay, that's what's happening right here. Okay, the oxygen accepts the electron and makes hydrogen. All right, I know that's difficult. Let's watch an animation to help us out. Yeah, I know, and that's why it's easy to get photosynthesis and respiration mixed up. That's completely understandable. Okay, let me show you this quick little animation.
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy, ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of two membranes, an outer membrane, separating it from the cytosol, and an inner membrane, surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes.
Complexes are the electron transport chain. This ATP synthase, you know what that is. That's what's going to make ATP. Your energy carriers are going to come in down here. Okay, so watch down here for the energy carriers. Okay, they're going to drop off electrons. The electrons are red. That's just to help you understand what you're watching.
Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix.
Look right here.
When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
All right.
Energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space.
Okay, a proton is just a hydrogen ion. Okay, protons and hydrogens are the same thing. And here we usually say hydrogens. Okay, watch right here. Watch this electron. Watch where it ends up.
Complexes to pump protons from the matrix to the intermembrane space.
Did you see what accepted that electron? Did you catch it? It happens real fast. The little glowy thing is the hydrogen. But watch right here for oxygen.
To the intermembrane space.
Okay, oxygen took that electron. Okay, but what this doesn't show real well is it also accepts two hydrogens. You can kind of tell because look, there's your two protons. Okay, so here's your hydrogen gradient. Okay, and it's going to move through ATP synthase. All right, watch right here. Okay, watch these. Hydrogen come in. Do you see how it's spinning? That's the motion that's needed to make ATP, and then ATP comes out. Okay, ATP is coming out. That's the red stuff. That green thing is actually a protein that you don't have to know the names of these proteins, but it's a protein that shuttles an electron. Okay, what you need to know is just the proton shuffle the electrons. So that's what I have written. You actually don't have anything to write down on these notes right here because I just put it all in. It was complicated enough. So I put your main points in for you. Okay, electrons are transferred from the energy carriers. That's your NADH and FADH+. Hydrogen accumulates. Oxygen is the final electron acceptor. And this is where I make my most ATP. 32 to 34 ATP are made in this step. All right, now we're going to do our summary, and we're going to use a silly analogy, and we'll be done. Silly analogy. Okay, this is your summary chart. If you can understand this chart, then that's pretty much everything you need to know about respiration. I'm going to write our formula down here. Whoops, that's photosynthesis. I'm going to write our formula so that we can come back and look at it. Okay, you don't have to. I'm just going to point it out. So if you can't write it, it's okay. This is just for me to come back and point some stuff out. Okay, I'm just going to put ATP for right now. Okay, so our first step is what? What is this first step? First thing I have to do is break down some glucose. What's that called? It's your first step. Glycolysis. It is oxidation of glucose, but the process is glycolysis. Okay, and I'm going to go from glucose to pyruvic acid. This does not happen in the mitochondria. It happens in the cytoplasm. Do you remember how many ATP are made? 2 little ATP. All right, we're going to fill this out, and then I'll come back with a little analogy to help you understand. Pyruvic acid ends up entering what? What's your next phase? Skip the link. Krebs. That's the next one you're responsible for knowing. And in Krebs, how many ATP do I make? 2 little ATP, and what else? Yeah, the energy carriers. NADH and FADH2. Okay, the way they're labeled up here is chemical energy. NADH, this says Krebs. All right, this right here at the top, it says chemical energy. I left that out on accident. You also make NADH in glycolysis. Okay, so that's what it means by high energy electrons. Okay, and then what's my final step when I make the most energy?
Spinny step.
The spinny step is the electron transport chain, or the ETC. Do you remember how many ATP I make? Yeah. It's 32 ATP. Okay, 32 to 34. Now, down here, in glycolysis, which reactant did I use? Glucose. Okay, now look at your products. I'm making ATP all along, but in the Krebs cycle, when I've broken down glucose as much as I can, I end up releasing a gas with carbon. Look at your products and tell me what gas is released. CO2 comes out. Okay, look at my products that I have left. Electron transport chain needs an electron carrier. Okay, or electron acceptor. What is that? Oxygen. It's the only thing I have left. And when oxygen accepts an electron in hydrogen, what comes out? Water. Hey, here's a silly way to remember this. Have you guys ever been to Chuck E. Cheese or an arcade? Okay, can you use your dollars? No, you got to get it converted, right? I think they use like a little wristband or a card now, but it used to be tokens. So this part, glycolysis, is like when you're standing at the entrance to Chuck E. Cheese, you got to give them your money and they give you tokens. That's glucose to pyruvic acid. Your dollars are glucose, your tokens are pyruvic acid. Okay, only when you've done that, only when you have your tokens can you go into the arcade. The mitochondria is the arcade. Okay, now you're going to play your game. The game is the Krebs cycle. As you're playing your game, what's building up? Points. Okay, you're building up points. Your points are your energy carriers. But you really don't want the points. You want what you can get with the points. What do the points translate into? Tickets, okay? So the ATP is your tickets. That's like the electron transport chain. Like when you hit the jackpot and all your points add up and it spits out a bunch of tickets to you, okay, your points are converted into tickets. The points are your electron carriers. Your tickets are your ATP. Okay, if that can kind of help you remember.